Hello, everyone, and welcome to today's webinar, Investigating Cell Surface Receptor Dimerization Using Single Molecule Super Resolution Microscopy. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you are having any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Katie Sherricks. Katie is a postdoctoral researcher at the Francis Crick Institute in London. Katie, you may now begin your presentation.
Thank you for the introduction and to Thermo Fisher for inviting me to do a webinar about some of my work. And today I'm going to be talking to you about some of my single molecule super resolution microscopy work looking at cell surface receptor dimerization. So this is just a disclaimer slide. And I want to start off by giving a little bit of an introduction about me and my background. So I did my undergraduate degree at the University of Sheffield, and I graduated in 2020 with an integrated master's in biochemistry and genetics. And in 2024, I finished my PhD, which was at Imperial College London, and I was investigating dopamine receptor dimerization, which is what I'm going to be mainly focusing my talk on today. And at the moment, I'm a postdoc researcher at the Francis Crick Institute, and I'm still working on membrane receptors, but in a project in collaboration with AstraZeneca. And I won't be talking about this work today, but just to highlight that my interests really lie in membrane protein receptor signaling and dimerization. And my work consists of a lot of cell culturing and microscopy, which is what I will be discussing today. Membrane protein receptors are a fundamental part of signal transduction pathways, and this means that they play a really key role in almost all physiological processes. And they crucially allow signals to pass across the cell membrane. And some of the key examples of this are the G protein coupled receptors and the receptor tyrosine kinases. And because they have a key role in many physiological processes, they are often altered in disease. And because of this, they have historically been highly tractable and successful drug targets, with GPCRs alone making up over a third of FDA-approved drug targets. So a little bit more about G protein coupled receptors. So these are a class of proteins that consist of over 800 members in mammals. And GPCRs are grouped into different groups according to their sequence homology. And the largest and most diverse group of receptors is the class A or rhodopsin-like receptors. And these are the targets of many different types of drugs and are also the focus of a lot of different research. So they're the most well-studied group. So receptors can form dimers or oligomers, and they can do this either with themselves or with other GPCRs to form homodimers or heterodimers. And some types of receptors depend on dimerization more than others. So, for example, the class C G protein coupled receptor family has to form dimers in order to function effectively. On the other hand, other receptors, such as this class A GPCR here, which is the beta-1 adrenergic receptor, doesn't have to dimerize in order to function, but has been shown to dimerize in order to diversify their signaling. And a number of different GPCRs have been shown to form dimers in physiologically relevant pathways, and some of these have a role in disease. Something that is important to note is that dimerization of G protein coupled receptors, and in particular class A GPCRs, is often very transient. So these can be difficult to capture these short-lived complexes through high-resolution structural techniques. So historically, many people have used biophysical assays such as Fret or Brett race assays to study receptor dimerization and oligomerization. And these are both proximity-based live cell assays, and they can determine whether a protein is within 10 nanometers of another protein. But what's important to note here is that these techniques cannot determine whether a receptor is located at the cell membrane or whether it is located intracellularly within the cell. And microscopy approaches ranging from fixed cells to live cell imaging have been used in recent years to more precisely measure receptor interactions within cells, including by only monitoring cells that are present on the plasma membrane. And these techniques include, but are not limited to, single particle tracking, fluorescence correlation spectroscopy, and single molecule localization microscopy. So today I'm going to be focusing on single molecule localization microscopy. And this is a group of techniques that is able to break the diffraction limit of light and achieve resolutions less than 200 nanometers, and even down to only a couple of nanometers resolution. So in microscopy terms, we think of resolution as the smallest distance between 2 points where they can still be seen as separate entities. And this is a limit of the microscope. So the lower the resolution, the greater your ability to see the precise detail of the molecules you are looking at. And this is particularly important when we're looking at things like membrane receptors, as these tend to be less than 10 nanometers in size. So in a diffraction-limited wide-field microscopy approach, such as conventional confocal microscopy, the resolution of these techniques is low. And you're unable to determine the precise localization of the fluorophore. You have this diffuse area where it could be, as I've shown in this figure on the left. There is a diffuse area where it could be. And this is what is known as the point spread function of an emitting fluorophore. And in these diffraction-limited techniques, such as confocal microscopy, you're unable to distinguish between two separate points, meaning the resolution is low. And this is because these point spread functions are overlapping. So single molecule localization microscopy techniques achieve this high resolution by breaking this diffraction limit and ensuring that point spread functions do not overlap. And this allows the precise localization of a fluorophore to be determined. And this is what I've indicated on these images as a cross. So you're able to determine the precise location rather than this diffuse area of a lower resolution image. So the most common types of single molecule localization microscopy are storm, palm, and paint. And what I will be talking about today mainly is palm. So generally, with these techniques, we achieve non-overlapping point spread functions by only activating a very small subset of fluorophores in each of the frames. And this ensures that we are temporally separating fluorophore emissions so that the point spread functions are not overlapping. And after imaging over many frames, the localization of all emitting fluorophores can be precisely determined. And from this, we can extract information such as dimerization by doing algorithms such as neighborhood analysis. So in this example here, where I've noted di or oligomer quantification, you would see that within a defined search radius that we call or R, in this example, the orange receptors here would be monomers, the blue ones would be dimers, and the green ones would be tetramers, as they are within a defined search radius of each other. So to focus a bit more on PD-Palm, which is the technique that our lab focuses on the most. So this stands for photoactivation localization microscopy with photoactivatable dyes. And what we do is we conjugate an antibody with a photoactivatable dye that is activated and excited in a stochastic manner. So in each frame, we only activate a very small subset of these conjugated antibodies. And this appears as blinking when we are imaging. And this is where you get excitation and emission of the fluorophore. And this is followed by bleaching. And if we conjugate the antibody and the dye in a 1-to-1 ratio, we can be sure that one blinking event and one localization is one receptor. So now that I've introduced single molecule localization microscopy as a technique to look at membrane protein receptor dimerization, I want to talk a bit more about my PhD work, which was looking at G2 dopamine G protein coupled receptor dimerization. So G2 dopamine receptors are class A GPCRs, and they have been implicated in a range of different diseases, in particular schizophrenia and Parkinson's disease. And they have been shown to form both transient homodimers with themselves and heterodimers with other GPCRs. And importantly, post-mortem schizophrenic brains have been shown to see an increase in D2 homodimers. So what we really wanted to look at with this project was to uncover the precise molecular interface between the two proteinomers or individual receptors within D2 homodimers. And the hope is that this information could be used to design better dimer-specific drugs. So D2 dopamine receptor signaling is mediated by G proteins and beta-arrestins. So this receptor usually couples to G-alpha I G proteins, which have an inhibitory effect on adenylyl cyclase, reducing cyclic AMP production in the cell. However, it also couples to beta-arrestin 2, which is an adapter protein that is known to act as a scaffold to recruit proteins to turn off signaling from the receptor. But it is also known to signal itself through pathways, including activating the MAP kinase pathway and mediating internalization of the receptor. And when we have equal signaling from G protein and beta-arrestin, we call this balanced signaling. So when we have a preference for one pathway over another, we have what we call bias signaling. So for example, preferring the beta-arrestin over the G protein pathway will produce more signaling effects downstream of beta-arrestin. And this has been shown to have effects in signaling and physiological or pathophysiological responses as well. So with this project, we wanted to first stabilize the receptor dimer. So as I mentioned previously, Class A GPCRs are often transient dimers, which makes them difficult to capture with high-resolution structural analysis. So in order to stabilize the receptor, we used a mutagenesis-based approach. And we identified residues based on computational molecular models of the D2 homodimer. And these residues were predicted to increase the dimer stability. So after making all these mutations and validating them, I wanted to look at how these mutant D2 receptors affect dimer stability. So I used a number of different techniques to assess this, including Western blot analysis, bioluminescence resonance energy transfer, and PD-PALM. So from this, a particular mutant really stood out, and this was the double V96S V97C mutant. And this is shown in the turquoise color here. So you can see that relative to wild type D2 receptor, you see an increase in relative dimer levels by Western blot analysis, an increase in Brett max, which indicates an increase in protemer proximity within the dimer with this mutant. And through PD-PALM, we showed that there was an increase in total oligomers in this double mutant expressing cells compared to wild type expressing cells. So since the focus of this talk is single molecule imaging, I want to talk a little bit more about the PD-PALM imaging in more detail. So we use photoactivatable KH500 dyes conjugated to an anti-flag antibody that recognize the flag-tagged receptor that is transiently transfected into HEC293 cells. So we did this with the wild type receptor and the mutant receptor to compare levels of dimerization in these transiently transfected cells. So PD-PALM imaging of fixed cells allowed a cell surface map of the D2 receptors on the surface of HEC293 cells to be determined. And neighborhood analysis allowed the oligomeric state of the receptor in these transfected cells to be determined. So what we were able to see from this data is that we saw an increase in total oligomers in HEC293 cells that were expressing the double mutant compared to those that were expressing the wild type receptor. And what's really good with PD-PALM is that you can look at the stoichiometry of the receptor. So you can see whether these receptors are made up of just dimers or trimers or higher order oligomers. And this can be particularly important for some receptors that may have differential effects depending on the complex that they are in. But what we saw in this particular case is that when you break down and look at what these oligomeric complexes are, there did not really seem to be any particular population that was enriched. So because these were transiently transfected cells, there were variation in the expression levels of the receptors. So we did also do controls looking at a range of different receptor densities as well. And we did show that these had an effect on the extent of dimerization, but didn't really influence the trends that we saw when comparing wild type receptor to mutant receptor. So I don't have time to go into detail about the signaling profiles of these mutant receptors today, but I want to highlight some of the key points here. So what we showed was that the signaling profile of D2 double mutant expressing HEC293 cells was altered compared to D2 wild type expressing cells. with a decrease in G protein-mediated signaling, enhanced basal beta-arrestin recruitment, and enhanced both basal and agonist-induced internalization. And this all suggests a switch towards more beta-arrestin-mediated pathways over GI. So we're seeing this signaling bias. And what was interesting is that we saw in the mutants that showed increased dimerization compared to wild type, we also saw altered signaling with a bias towards beta-arrestin. And to investigate this theory a bit further, our collaborator carried out molecular modeling with the predicted D2 homodimer in complex with either beta-arrestin 2, which is shown on the left-hand side, or with G protein, shown on the right-hand side. And this revealed that when you have a dimer of the receptor, it can accommodate 2 beta-arrestin molecules. However, when it's in a dimer, steric clashes mean that only 1 G protein molecule combined per dimer. So this provides some explanation for the observed effects of receptor mutants, which showed an increased dimerization and also showed a bias towards beta-arrestin 2 associations. As within a dimer, you've got more space for more beta-arrestins to bind than G protein. So it makes sense that you would therefore get more signaling of beta-arrestin mediated pathways. So in conclusion, this study found that using molecular models of D2 dimers, we were able to identify stabilizing mutations, which increase the stability of the D2 dimers relative to wild type. And importantly, one of these receptors, which I focused on today, the double mutant, showed an increased propensity to form oligomers through a range of different techniques, including PD-PALM. And overall, this more generally suggests that D2 dimers may favor associations with beta-arrestin 2. So on the left-hand side of this diagram here, I've shown what the wild-type receptor tends to do. And then on the right-hand side, I've shown what this double mutant seems to do. So we see increased dimerization, increased associations of beta-arrestin in the absence of any ligand, weaker G protein binding, beta-arrestin dependent downstream signaling activation, and an increase in internalization. And to summarize overall why you would use single molecule super resolution microscopy to study membrane protein receptor dimerization. So we're able to quantify receptors at the plasma membrane specifically, and this can really be complemented with other techniques such as the resonance energy transfer and Western blot to ensure that you are looking at associations happening within the plasma membrane that you're interested in. And this allows single molecule resolution of receptor dye and oligomers. And PD-PALM specifically allows you to determine the receptor complex stoichiometry. And this could have really important effects, whether you're looking at dimers or bigger complexes. These might have distinct effects. And you wouldn't be able to determine this through bulk assays such as resonance energy transfer. So I would like to thank the co-authors of the paper of the study that I presented here today from Imperial College London, and our collaborator, Francesca Finelli, who did the modelling that I presented here. And I'd also like to acknowledge my current position, even though I didn't show any of the work today. I'm still working on PD-PALM to look at receptors, but a different type of receptors, looking at receptor tyrosine kinases this time. And here are my contact details in case you want to get in contact. My email, my social media, where I I really try to show what it's like working in academia in a transparent way, and also do a little bit of science communication, so feel free to add me on there. And thank you everyone for joining this webinar.
All right. Thank you very much, Katie, for your very informative presentation. We will now move into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. All right, let's go ahead and get started here. The first question that we have says, how are you able to determine which is the best super resolution microscopy approach for your application?
Yeah, so I think it really depends on what you want to look at and what your focus is on. So, for example, what type of cells you're using or what type of material you're using for the microscopy. So things like fixed cells or live cells or adherent cells or tissue samples. Different microscopy techniques tend to favor these different sample types more or less. And one of the main things is you want to decide whether you want high spatial resolution or high temporal resolution. So do you want a really high resolution image, or do you want to know about the kinetics and things like that? So in reality, it might be a combination of different techniques. So things like looking at, for example, PD palm to get the high spatial resolution, but then combining that with something like single particle tracking, so you get more of the dynamics as well.
All right, fantastic. Thank you. The next question that we have here says, what could be the therapeutic implications of the biased signaling of D2R dimers?
Yeah, so we know that in post-mortem schizophrenic brains, there is an increase in the D2 dimers. So what this study showed was that there is a relationship between D2 dimers and association with beta-arrestin. So it might be that with these specific mutations, we're locking the receptor in a more active-like conformation. And if we want to target D2 dimers specifically, we need to be aware of what signaling implications that might have. So, for example, breaking apart the D2 dimer might switch the signaling back to a more balanced signaling away from this bias. So I think really exploiting the dimer and using that to change the signaling would be where therapeutic approaches would go.
Perfect. The next one that we have here says, how do transiently transfected HEC293 cells compare to what would be seen in the brain in terms of expression or receptor density?
Yeah, so with transiently transfected cells, within different transfections and also within the same transfection, we were getting different receptor density and different cells. So we did image across quite a range of different cells of different receptor density. And within the analysis, we're able to determine the total receptor number. So we can say whether it's a high density or a lower density. And we did find that receptor density had an effect on the proportion of the receptors that were in a monomeric complex. But regardless of whether you had a low receptor density or a higher receptor density, the trends we saw were the same. So we still saw this difference between wild type and the mutant. So it seems to be, at least the trends we're seeing seem to be receptor independent. And sometimes people say, oh, if you've got high receptor density, you're not just forcing the receptors together. But we saw, even at lower receptor density, we still saw dimers. And in the brain, the density of the receptors tends to be quite high. So if anything, with this technique, we're actually underestimating rather than overestimating the dimerization oligomerization.
Okay, great. Thank you. The next question that we have here says, what are the main advantages or disadvantages of PD palm?
Yeah, so I think the main disadvantage, as I sort of talked about in a previous answer is that with this technique, you're only getting really a snapshot because they're fixed cells. So you might miss some of the dynamics of the interactions between the receptors. But what's really powerful, particularly with PD-PALM, is that you're able to determine the stoichiometry of the complex. So you can say whether it is in a dimer, a trimer, or something else. And with certain receptors, we know that the type of complex can influence how it signals. So looking at one type of receptor complex might be really useful for this.
All right. We have a couple more that have come in here. We have a comment that says, thanks for the opportunity to watch this webinar and learn something new. The next question that we have says, how does the cell decide which pathway they have to follow to make homodimer or heterodimer in terms of cell surface receptors?
So that's something we don't actually understand that well. So it's likely that this is dependent on the context, so dependent on the cell context. So things like different ligands, so ligand-dependent formation of receptor complexes, so homodimers or heterodimers, it can be influenced by the type of ligand. So one ligand you might favor a homodimer complex, another ligand might favor a heterodimer complex. And it's also likely cell type dependent and dependent on the receptor, the density of other receptors. So, for example, we know that in the brain, different brain regions, you tend to form either more homodimers or more heterodimers, depending on what other receptors are there and how highly they're expressed.
All right, fantastic. We have another here. It looks like this is a two-part question. It says, I was wondering, in the context of drug interactions, how can this technique be applied to monitor drug-induced receptor dimerization at the cell surface? And then the second part says, and what kind of insights can it provide into mechanisms of action or resistance, particularly in the case of targeted therapies?
Yeah, so what I've touched upon a little bit is how we can really exploit what we know about dimers for drug interactions and designing specific drugs that might target one complex over another. So what we could do, and what I didn't go into too much, is that we can do this technique. We can treat the cells or whatever sample you're looking at with specific drugs and then monitor before drug addition and then after, and see how that changes the dimerization. So that would be one way you could look at that. And to answer the second part of your question, to do with the mechanisms of action, really understanding the downstream implications of this would be really useful. So things like if we, using this microscopy, if we saw that treating the cells with a specific drug causes changes in dimerization, we can look at how treating the cells with this drug affects signaling. So things like the beta-arrestin assays or the G protein assays that I was talking about before.
All right, wonderful. It looks like we have another question here that has two parts as well. This one says, thank you for this insightful presentation. I have a couple of questions. How do you distinguish between real dimers and random in your analysis? Are there technical limitations for studying dimers in live cells versus fixed samples?
Yeah, so to answer the first part of your question, generally this is controlled for by doing controls. So things like doing negative controls so we can monitor what the background level is. When we have, for example, no antibody on there or no cells in the dish, you can monitor the background that way. And then we have, within the analysis pipeline, we have filtering parameters, which we have optimized so that we are sure that what we're counting is real interactions. And then the second part of the question, so to answer it, basically, yes, there are technical limitations. So it's a technical limitation based on the technique. So for PD palm, because we are looking, you're imaging over a time course, so you will add your conjugated antibody for the for PD palm specifically. You add your conjugated antibody to your fixed cells, and then you will image for example, 10 minutes. And then because we're stochastically activating only a few receptors at the time, we will build an image of that fixed sample. So we're not able to do this in live cells because then the image that we're looking at will change. So we're not able to build this detailed high resolution image. Hopefully that answers your question.
All right, wonderful. So we will go ahead and wrap up with this final question that we have here. It says, since your study focuses on palm, could you share what advantages it had over other super resolution techniques like storm or paint for your specific application?
Yeah, so the main differences between palm and storm is that palm uses photoactivatable dyes, whereas storm uses a oxidation reduction buffer system. So really it was easier for our applications to use the PD palm technology, as the storm protocol often requires you to optimize the buffer solution. And with with DNA paint, with that technique, you are not able to determine the stoichiometry of the complexes like you are with PD palm. And we really wanted to make sure that we were looking at specific populations, so either dimer or higher order oligomers. And with DNA paint, you're not able to determine that. It's just bulk receptor on its own or receptor in an oligomeric complex. So that was our rationale for choosing that particular technique.
All right, fantastic. Well, thank you again, Katie, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thanks everybody. Goodbye.